So how do we increase substrate for this reaction? So during the onset of exercise, we have ATP being used by various sources that we covered last class to allow for contraction. So we have sodium-potassium pumps on the membrane that open to allow membrane depolarization and axial potentials to be conducted across membrane. We have myosin ATPases that perform power stroke to pull actin shortened muscle. And as our muscle undergoes repeated contraction relaxation phases, we have our sarcopumps that pumps calcium back into the sarcoplasmic reticulum. So this process break down ATP to perform work, and they break high energy phosphate bond, which is the phosphate group. Therefore, the phosphate group is cleaved, and we get ADP and work. So, for example, the work would be contraction of the muscle. Once ADP is formed, so once this reaction happens, ADP form serves as a substrate for the creatine kinase reactions. So according to the law of mass action, if you increase substrate, you drive the reaction forward. So breakdown of ATP due to muscular contractions increases ADP, which drives the creatine kinase reaction forward. Therefore, ADP combines with phosphate group in the phosphocreatine molecule to produce ATP and creatine. So we can see the effect of substrate. So if we have low substrate, we're gonna have low products, a low amount of products. However, as soon as we increase the substrate amount, we're gonna increase the amount of products. So by altering the concentrations of substrate, we alter the pressure gradient of this flow model and drive reaction forward. Theoretically, we could also drive this reaction forward or increase flow by increasing conductance. However, in this case, the rate of ATP production is influenced primarily by the availability of substrate. So the amount of ATP and phosphocreatine present within the muscle is governed primarily by the law of mass action. So as we go through different ATP producing pathways in muscle, we will add them to our metabolic map. So our first ATP producing pathway in muscle is the phosphocreatine system, which results in one molecule of ATP formed for each molecule of phosphocreatine that is consumed. The next anaerobic pathway we will cover is glycolysis, which occurs in the cytosol. And the glyco is relating to glucose, and the lysis part of glycolysis, it means the breakdown of this process. So again, anaerobic does not require presence of oxygen. Remember, anaerobic does not requires oxygen for the reactions. So in this case, glycogen is the storage form of glucose in muscle, can be broken down when needed. Alternatively, can take up glucose directly from blood to break it down and convert it into energy right away. Both glucose and glycogen undergo glycolysis. We'll look at how this pathway, the amount of ATP produced, differs depending on glucose versus glycogen as our starting the substrate. So both glucose and glycogen must be converted to glucose 6-phosphatase. in addition to which is an addition. So both glucose and glycogen must be converted to glucose 6 phosphate, which is the addition of phosphate group, before they can enter the glycolysis pathway. The key difference between using glucose versus glycogen as the starting point for glycolysis is that conversion of glucose to glucose 6 phosphate requires energy. So one ATP molecule is consumed. When glycogen is a starting point for glycolysis, its conversion to glucose 6 phosphate does not require energy. Rather, it binds to an inorganic phosphate and is converted to glucose 6 phosphate. So we save one ATP when starting with glycogen as our substrate. It's also important to highlight that the enzymes that are involved in the system. So we have hexokinase, which catalyzes the conversion of glucose to glucose 6 phosphate. And remember, this reaction consumes one ATP molecule. We have glycogen phosphorylase, which catalyzes the conversion of glycogen to glucose 6 phosphate. And this reaction does not require ATP. So let's start with the investment phase. So starting with either glucose or glycogen, and the product of this pathway is pyruvate. So the first thing we do to understand this process is divide into investment phase, which is the ATP consuming phase, and the production phase, which is the ATP producing phase. So in the investment phase, 2 ATPs are consumed when starting from glucose, and when starting from glycogen, we save 1 ATP. So starting with either glucose or glycogen, the product of this pathway is pyruvate. So the first thing to do to understand this the glycolysis is to divide into so starting with either glucose or glycogen, the product of this pathway is pyruvate. So the first thing to understand glycolysis is to divide the glycolysis in 2 phases. So we have the investment phase, which is ATP consuming phase, and we have the production phase, which is the ATP producing phase. In the investment phase, 2 ATP are consumed when starting from glucose, but when starting from glycogen, we save 1 ATP. In the production phase, this phase occurs twice per molecule of glucose or glycogen. So we have 2 ATP produced per phase, and that happens twice. So we have 4 ATP produced altogether. Also, this phase also produces NADH, which is an electron carrier that transfers electron to the electron transport chain. And we'll look at this later. So the key points here is that in the investment phase, we consume 2 ATP when starting from glucose, and when starting from glycogen, we save 1 ATP. In the production phase, we have 2 ATPs being produced, 1 NADH, but since this happens twice, we have a total of 4 ATP being produced, and 2-NADH being produced from glucose or glycogen. So the net production per turn of glycolysis is 2 ATP when starting from glucose, because we lose 2 in the investment phase and we gain 4 in the production phase, and 3 ATP when starting from glycogen, because we don't lose that extra ATP in the investment phase. And the last one is 2-NADH from glucose or glycogen. So the important enzymes of glycolysis are hexokinase, glycogen phosphorylase, and phosphofructokinase. They are the most important enzymes of the glycolysis. Hexokinase converts glucose to glucose 6-phosphate and consumes 1 ATP. The glycogen phosphorylase converts glycogen to glucose 6-phosphate and does not consume ATP. And we have phosphofructokinase that converts fructose 6-phosphate into fructose 1,6-diphosphate and consumes ATP. The 2 ATP consumed in the investment phase are due to hexokinase and phosphofructokinase, because glycogen phosphorylase does not use ATP. So this is why when we start off with glycogen, we save 1 ATP. So the glycolysis regulation, we have 3 important factors in order to glycolysis to happen. So we have the concentrations of key glycolytic enzymes, which are hexokinase, glycogen phosphorylase, and phosphofructokinase. Another factor is the levels of substrate, either glucose or glycogen, and oxygen. The abundance of oxygen inhibits glycolysis. So in the initial phase of glycolysis, the investment of ATP is 2 ATP for glucose and 1 ATP for glycogen. In glycolysis breaks down glucose or glycogen. So in the producing phase, this phase happens twice for each glucose 6-phosphate. So in the second phase, we have the production of 4 ATP for each molecule of glucose 6-phosphate. Therefore, increasing glycolytic flux will increase in flow for ATP. So you don't need to remember every single step of the glycolysis, but it's extremely important for you to remember this, the summary of the amount of ATP produced in the glycolysis. So in the investment phase, we have the use of 2 ATP for glucose and 1 ATP for glycogen. And in the production phase, which occurs twice, we have 4 ATP being produced from glucose, 4 ATPs being produced from glycogen, and 2 NADH being produced in this phase. And this is occurring twice. So the net production will be 2 ATP from glucose, 3 ATP from glycogen, and 2 NADH. So that's the summary for glycolysis. So now we can add this second part in our ATP producing pathways. Now we can add glycolysis into our map. So remember, we have either glycogen or glucose being broken down by either glucose phosphorylase or hexokinase or phosphofructokinase. Then we have the production of ATP and NADH. And the final